This is a brain. It is estimated that brains contain 85 billion neurons. The neuron is a nerve cell and is the primary functional unit of the nervous system. But how do neurons relay information to the next neuron? The neuron uses electrical impulses and chemical signals to transmit information across the brain. What you often see regarding signal relay is that at the synaptic cleft, the neurotransmitters from the presynaptic side will be released from calcium-coated vesicles for the corresponding receptors to receive on the postsynaptic neuron. This will pass the information along. However, the story of signal relay is a little bit more complicated than that. In recent years, there are proteins that have been identified to reside on the surface of these synapses. Let's take a look at cadherins, for example. Cadherin stands for calcium-dependent adhesion proteins. There are type 1 transmembrane proteins important in the formation of adherent junctions for neurons to bind to each other. Complications caused by these adhesion proteins may lead to many different types of inherited disorders in humans, including skin and hair disorders, cardiomyopathies, sensory defects associated with deafness and blindness, and psychiatric disorders. In the Brasch Lab at the University of Utah, we use techniques such as X-ray crystallography, cryo-electron microscopy cryo-EM, and cryo-electron tomography cryo-ET to visualize proteins at the synaptic cleft to see how they work together. From visualization of the synaptic cleft, there are different types of interactions that synaptic proteins are able to form. The stronger type, trans interactions, where these proteins will interact with each other from different neurons, or the weaker cis interactions, where proteins will interact with each other from the same neuron. They can also bind to proteins identical to themselves, and this is called homophilic binding. In other cases, they can bind to a different protein or many different types of proteins, and this is called heterophilic binding. Learn more about us in the upcoming videos on more details about synaptic adhesion proteins and overviews of visualization techniques mentioned in this video.